Hello friends, welcome back to the channel. We are going to explore one of the most basic but very important topic of biology. Since the discovery of cell by Robert Hooke, we all know that it is the building block of all living organisms. Among cells, most important activities are its interactions with the environment. A constant, a continuous give and take that never ceases. And the one that is responsible is the delicate skin of lipid embedded with protein that encase every living cell and eukaryotic organelles. It is called the plasma membrane. And today we are going to learn about its structure and function. So let's begin. Right. The structure of cell membrane is called fluid mosaic model, which was proposed by Singer and Nicholson. This is a modification of the previous known model called the sandwich model given by Danieli and Devson. According to the fluid mosaic model, proteins can be found embedded within lipid bilayer, where the proteins can be seen floating freely in the lipid matrix representing boat on a lake. Now this is all possible due to the nature of the lipid bilayer. The lipid is mainly phospholipid, which has two parts, the polar phosphatidylglycerol head and the non-polar long-chain fatty acid tail. The head being polar can form hydrogen bond with water, whereas the tail they cannot. This results in formation of the head as a hydrophilic end and the tail as a hydrophobic end. The hydrophilic head region has an affinity towards water and the hydrophobic tail does not. It just moves away from water, resulting in forming a lipid bilayer. Due to this reason, in a water-filled environment like the extracellular medium and the cytoplasm, we find that the lipid bilayer is formed as the hydrophilic head region remains towards it, whereas the non-polar tail region remains away from it. Next component of the cell will be the protein, which can be said as the major component of every membrane. Some can be found as freely floating, whereas some can be found bound across, like this too. So what we will see here is that the transmembrane protein of this two kind, they have a nature of allowing movement of particle across, where this one has actually the property of letting the particle move across it, whereas this, it serves a complete different purpose, mostly recognition. What we will find here is that this forms a channel across the entire membrane, as the membrane being of non-polar interior, they will not allow polar particles to move across it, whereas this actually allow the movement of any polar particle across it. Next major component of the cell can be called these two. They are carbohydrates. The carbohydrates which are bound to the protein are called glycoproteins, and the one bound to the lipid will be called glycolipids. Well, their major purpose is recognition. We will come to that later on. Other components that can be found will be, surprisingly, the cholesterol molecules which are bound in the membrane and this cytoskeleton membranes materials that are there in the inner surface. Cholesterol mainly helps in maintaining fluidity, whereas the cytoskeleton, as the name suggests, it helps in maintaining shape and integrity of the cell. Yes, there are some inert proteins that can also be seen here. The inert protein is mostly serving the purpose of signal transduction, which is a way of communication across the cell. Say this cell communicating with something other or another cell, or maybe with the environment. So the signal can be passed down by means of this inert protein. We'll come to that again later. So when we talk about all of this component, this is the major component that can be found. Sometimes additional to this, some other forms of protein, some other things can be seen here, whereas mostly in terms of functionality, these are the ones that will be there. Now let's come back to function of the components. So when we sort them out, we can say the components and their functions are diverse. Let's pick up a topic. Let's say the lipid bilayer. When we talk about the lipid bilayer, its major function will be it being a fluid matrix. Being a fluid matrix, it allows protein to be embedded within, or the one which are already embedded within, to move across it. And also it is very flexible. But due to its nature, it does not allow permeability to every material, like for example polar substances, they cannot move across, or any charged ion, they cannot move across. So they will be all bound to the outer surface but will not move across the membrane. This results in the membrane being selectively permeable. It will allow movement of non-polar substances but will not allow movement of any other particles which are polar or which are a bit charged. For example, let's say if anything which is fat soluble, say example the vitamin A, which are known as fat soluble, they can move across it, but other substances will not. Another thing, oxygen, carbon dioxide, they also can move across the membrane without any problem because they will be diffused easily. Whereas any other charged particles like sodium ion, potassium ion, they will not be allowed to move across. Next component, surprisingly, the cholesterol will actually very important. People think cholesterol is not present in the membrane, but in reality they can be found mostly in the animal cells, and their main purpose is to dampen any effect of temperature. Because if there is a rise or fall in temperature, the fluidity of the membrane actually it changes, and the cholesterol, it dampens the entire effect, resulting in maintaining fluidity of the membrane. So we can write cholesterol, its function is maintaining membrane fluidity as it dampens effect of temperature. Next component will be the transmembrane proteins. Now transmembrane proteins, they are mostly of two types. They are the carrier or the channel type. I'm sorry, I was off focus. They are the carrier or the channel type, and their main purpose is to transport materials across. This is type A. See, there are active transport system and there are passive transport system. They are mostly run by the carrier or the channel proteins, which mainly being a part of the membrane, allow the particle to move across it. Whereas membrane proteins like the receptor types, they actually helps in interaction or transmission, or let's write transmit signals. For example, hormone receptors. So by the name you can only guess that these are the receptor proteins or the proteins which will carry a signal which has come to it, say the hormone, and will transmit the signal to the cell so that the cell can work accordingly. Along with the receptor protein actually works the interior protein, which as I said helps in signal transduction, isn't it? Now signal transduction is actually a process in which the signal that it has received, say the hormone signal, it will carry the signal and pass it along to the nucleus. For the nucleus, until and unless it receives the signal, it cannot work. So the signal transaction is a very crucial thing for every single cell, be that a plant or be that animal, every single cell. And this signal transaction is mainly carried out by the interior proteins that can be found embedded as like this one. So example can be Janus kinase. Alongside with this, there are proteins like, for example, the spectrins, or say tubulins. So they help forming the cytoskeleton. They are cytoskeleton. So just integrity of the cell. Finally, we can say there are two most important things, which are the cell surface markers. For example, the glycoproteins. The glycoproteins are the one which has a very important task of self recognition. Self-recognition. Now what does this word self-recognition means? It actually means now this glycoprotein will help the immune system of our body to recognize whether the cell is of the normal type or the one which has turned cancerous. So it can sort it out and can work accordingly. For instance, there are specific cells like the cytotoxic cells, the cytotoxic T lymphocyte or the CTL, they are the one which will attack and will kill the cancerous cell, whereas the normal cell will be saved. Now this is all possible due to a specific molecule called the MHC complex, or its full name is major histocompatibility complex, which helps in the recognition process, and they are nothing but glycoproteins. Finally comes the glycolipids. Now they also help in recognition, but the recognition type is different. This is mostly tissue recognition. Now what does it mean? Well, simply one way answer. A, B, O, blood type. Now we all are familiar with the ABO blood type. Now it is nothing but a small carbohydrate molecules that can be found bound on the surface of the RBC, which helps in recognition of the RBC as type A or type B or type O or maybe type AB, depending on which particular set of carbohydrates are present on the surface. Now this helps in recognition of the blood type. Now this is one example. Nearly every single type of tissue can be recognized simply by seeing the type of glycolipids that are present on top. Now these all are the different part and its function. Now when we see them together, for example, let's say the lipid bilayer, the cholesterol and the transmembrane proteins, we can clearly see that they help in maintaining the integrity of the cell, means to maintain the shape, and also it helps in exchange of materials with the cell with the environment. For instance, it helps in maintaining the shape as they are working together, plus it is helping in movement of particles across. So that way it helps in maintaining its shape, and also it helps in exchanging the materials with the cell and the environment. But at the same time, the proteins and the cell surface marker, say the interior protein also, they all help in recognition of the cell and the surrounding. It helps in interaction of the cell with the surrounding. It helps the cell to be recognized by a different cell type, maybe, let's say, or the immune system, let's say. Now that way we can easily see that the membrane becomes a very crucial component of cell, and without it the integrity, the function of the cell cannot be properly maintained. I hope that was informative. Thank you for watching. Please wait until the next video.